Assalamualaikum warahmatullahi wabarakatuh.
Good morning and best wishes for all of us.
The Honorable the Board of Jury of Indonesian Science Project Olympiad 2022.
Wilson's disease is a genetic disease that causes damage to the liver and or brain. This damage occurs due to the accumulation of excess copper in the body. This disease is rare but deadly.
It is not easy to detect someone who has Wilson disease because the symptoms and signs are similar to liver disease or other neurological diseases. Even though, through early detection and treatment, death from this disease can be prevented. However, the detection of this disease is still very minimal and expensive.
This made us to find a solution to this problem. Finally, I thought of using the purple golden flower that grows a lot in Probolinggo City where I live. Because of the belief in the function and the minimum use of this purple golden flower, it makes us want to take advantage and look deeper to solve this problem. Together with my friend, we proceeded to consult with the supervisor.
Based on the result of the discussion and also suggestion from the supervisor, finally, we present a research as well as become our scientific paper with the title Kategori Ruellia Wilson Test Kit based on the artificial matrix test from the combination reaction of normal urine and copper sulfate.
I'm Amalyah Nabita Ramadhani.
And I'm Cristian. We are students of SMA Negeri 2 Probolinggo.
Based on the problems, we set the focus of the problem formulation are: first, how to make Kategori Ruellia Wilson's test kit as a Wilson's disease detector from purple golden flower petals, Ruellia tuberosa L.
Second, how is the effectiveness of Kategori Ruellia Wilson test kit on the quality of Wilson disease detection result through a replacement matrix for the combination of normal urine and copper sulfate.
Third, how does Kategori Ruellia Wilson's test kit work in detecting Wilson's disease through a replacement matrix for a combination of normal urine and copper sulfate? Here is our research method. We conducted this research from November 1st, 2021 to January 30, 2022 at the Biology Laboratory of SMA Negeri 2 Probolinggo. The FTIR test for the compounds contained in Kategori and the reaction results was carried out on January 25, 2022 at the Laboratory of Minerals and Materials Central Laboratory in State University of Malang.
The following are the research variables that we use. It can be seen that there are 4 treatment variables. Namely, treatment A is Kategori using distilled water. Treatment B is a Wilson disease urine matrix. Treatment A3 is a treatment to test the reactivity of Kategori on Wilson disease matrix. And treatment A4 is a treatment for test the stability of Kategori on Wilson disease matrix. The variation in treatment combination can be seen in the following slide.
The following is a brief procedure of our research. First, purple kencana flower compound extraction procedure. Second, Wilson's disease urine matrix manufacturing procedure. Third, testing application procedure Kategori. Fourth, quality testing procedure of test kit Kategori.
Point 1. Kategori reaction with Wilson disease matrix test result. Mixing of Kategori with Wilson disease matrix or urine mixture with copper to sulfate solution before heating shows a purplish brown color as shown in the following slide. However, when heated, the solution will gradually change color to a green brown color and a dark green sand-like precipitate appears at the bottom. All treatment A1 to A8 showed almost the same result.
Point 2. Kategori stability test results. The following are the results of stability test for each treatment. It can be seen the higher the concentration of copper content, the more deposits or lumps formed in the darker the color. So, the stability test has an effect on the amount of clumps and lightness of the resulting color. Kategori reactivity test results. To improve the accuracy of Kategori, a reactivity test has carried out under certain conditions. The conditions we use today focus on acidic and basic conditions. Dear jury, the Kategori reactivity test shows a very interesting thing. Apparently, when Kategori is mixed with an acid solution, it will change color into a purplish or magenta. However, when reacted with an alkaline solution, it will turn orange. It is also unaffected or constant before and after heating. This event shows that Kategori liquid is very stable and accurate for detecting copper compounds.
Point 4. FTIR spectra IR test result. It can be seen below is the comparison of IR spectra between thicker and liquid and solid reaction products. It can be seen that the graph on the left contains CO groups of aromatic ester, aromatic CO2, and aromatic CH, which are not present in the graph on the right. However, the graph on the right has NX amines group, NX aliphatic primary amines, and CH alkenes, which are not present in the graph on the left. This indicates that the LS organic compounds detect the presence of copper compounds by binding to the functional group.
The following is a discussion for our research. The red pigment color of purple golden flower petals is thought to be malfidine, which is one type of anthocyanin member. According to Bagat, in 2008, stated that the anthocyanin of purple golden flower in its structure contains flavinium cations. Color changes can occur due to changes in the shape of the structure caused by the influence of pH. This shows the characteristic of the color change that is formed from the characteristic of the carbon. As for the precipitation that occurs in the reaction, the copper returns to anhydrous form as in the following reaction. It can be seen that the heating results make the copper saturated and deform into tetrachloropyr precipitate.
The conclusion of this study is that based on the concentration of the result of the study, it was determined that all treatment with highest concentration or concentration of 25% showed significant color changes and different characteristics. The following is documentation of our research activities. That is all from us. Ticker 1. Stop using this early for healthy life.